So here is the handout for today. These handouts, I continue to post these on Canvas file. You can find links for these on the wiki page called Lecture Notes Horvath. And for today, I've chosen a few images from the text and drawings that I've made myself to highlight features for the myosin ATP ACE domain and how that system couples ATP hydrolysis to molecular motion, aspects of which are illustrated here, and then also the ATP synthase system. And you'll see that I've drawn the same images for the first two boxes for both myosin and ATP synthase. And that's because the chemistry is highly analogous and comparable. The nucleophile and leaving group are different, but the transition state is exactly the same. We have this pentacoordinate phosphorus transition state. On this portion is ADP, and over here is water. And for myosin, water is the nucleophile, ADP is the leaving group. When we're fabricating ATP at the catalytic subunit of ATP synthase, now ADP is the nucleophile and water is the leaving group. But in both cases, we encounter this pentacoordinate, it's also called the pentavalent phosphorus transition state. The types of molecular motion that are coupled to the formation of this transition state are different. For myosin, we see that ATP hydrolysis drives the walking movement of myosin along a track of actin. And for ATP synthase, it's a circular type of motion driven by the movement of protons across the mitochondrial membrane. And this circular motion provides the mechanical force needed to fabricate ATP. For both myosin and ATP synthase, and also several other proteins, they all belong to this superfamily of proteins that contain a P-loop NTPase motif. And here are three examples from the textbook. Here is the beta subunit of ATP synthase. This is where ATP is being fabricated. Over here is the alpha subunit of transducin. You'll recognize this from the G protein coupled receptors. This is a place where GTP is hydrolyzed, and that's important for making sure that there's an off button for these cell signaling systems. And then myosin also contains, as part of its ATPase domain, a P-loop, and this P-loop, it's the details of stabilizing this transition state are linked to movements in the so-called switch loops, and then these switch loop movements are amplified to a degree in the movement of a relay helix, and then this relay helix movement is further substantially amplified into the movement of this lever arm. And this lever arm movement is what explains this really dramatic motion of the motor along actin filaments.